Hello, my name is Kate Campbell, and the bacteria that I will be discussing in this video is Campylobacter jejuni, or what I will be referring to as C. jejuni. This is a bacteria with a taxonomic status as follows. Phylum Protobacteria, class Epsilon Protobacteria, order Campylobacterialis, family Campylobacteriaceae, genus Campylobacter, and lastly its species names, which is the one that it was introduced as, Campylobacter jejuni. Other bacteria in the genus Campylobacter include Campylobacter coli, Campylobacter fetus, and Campylobacter leri. However, Campylobacter jejuni is by far the most common type of bacteria in this genus involved in human illness. This bacteria is listed under food microbiology, but based on certain topics touched on in this video, it will be clear that it can pertain to other categories of microbiology as well, such as agriculture, environmental, and human medicine in some way. Morphology. Getting into the cellular morphology of this bacteria, what does it look like? C. jejuni is a motile spirillum-shaped gram-negative bacteria. It can be anywhere from 0.2 to 0.9 micrometers wide and 0.05 to 5 micrometers in length. A distinguishing feature, apart from its spiral shape, is the polar flagella, found at one or both ends of the bacteria. Both of these features aid in motility. The spiral shape allows a corkscrew-like motion in media, and the flagella help propel the bacteria. C. jejuni is classified as a microaerophilic bacteria. This means they require oxygen to grow, but very low concentrations of it. Another feature to note is that they do not produce endospores. This means they won't be able to withstand harsh conditions out of their optimal living conditions. With an optimum growth temperature of around 37 to 42 degrees Celsius, they are thermophiles. With being microaerophilic and lacking the production of endospores, C. jejuni will be killed when exposed to hot temperatures and in the presence of high oxygen concentration. With old cultures or ones exposed to air for long periods of time, the physical shape of this bacteria will change to coccoid. However, this means that the bacteria has lost its motility and ability to transmit or colonize. With these features making this a delicate bacteria, Mueller-Hinton broth, or agar, supports the best growth of C. jejuni when incubated at 37 to 42 degrees Celsius. This bacteria is also classified as a risk group 2, as they pose a moderate risk to individuals but a low risk to the community. Proper level 2 lab containment is required to maintain safe lab protocol when working with this specific bacteria. C. jejuni colonize at the surface of mucous membranes of the alimentary, which is the digestive tract, or reproductive tracts. This is made possible by the adaptation in their morphology with their spiral shape and polar flagella. These allow the organisms to use their corkscrew motion to break through the mucus which other bacteria has a hard time getting to. C. jejuni is non-saccharolytic, meaning it cannot use glucose as a carbon source. It also is inhibited by high oxygen concentrations due to its microaerophilic nature. They can utilize various electron acceptors for respiration, including small amounts of oxygen, fumarate, nitrate, and in some cases, even hydrogen. C. jejuni uses specific amino acids such as aspartate, glutamate, serine, and proline, and intermediates of the citric acid cycle to efficiently generate energy, maintain growth, and persistently survive in certain environments. Some strains of C. jejuni have additional pathways that let them use glutamine and asparagine for their metabolic needs. Genome size. The genome size of C. jejuni is 1.69 million base pairs, has a circular chromosome, and it codes for approximately 1,654 proteins. The genome of C. jejuni is unique because it has almost zero insertion sequences, phage-related sequences, or repeat sequences. One of the most surprising discoveries was the presence of hypervariable sequences. These are short stretches of the same nucleotide, repeated, and are often found in genes that make or modify surface structures, or in nearby genes with unclear functions. The high variation rate of these repeating sequences may play a key role in how C. jejuni survives. All strains of C. jejuni have a gene for cytolethal distending toxin, but not all produce it. This toxin causes DNA damage, stops cell division, and leads to cell death in host cells. It is a virulence factor, meaning it helps the bacteria cause disease. It plays a key role in the development of illnesses caused by C. jejuni. Where can this bacteria be found? C. jejuni is most commonly found in the intestines of animals. Some include poultry, cattle, swine, rodents, wild birds, and even potentially household pets such as cats and dogs. It can also be found in untreated water, and this is usually caused by contaminated fecal matter in the environment, usually manure. What is the harm of this bacteria? Exposure to C. jejuni can lead to contracting Campylobacteriosis, which is a diarrhea-inducing foodborne illness. It can be contracted through a list of things, but common sources include raw or undercooked meat like poultry, beef, pork, and lamb, raw milk or other raw dairy products, raw vegetables that have come in contact with contaminated manure, shellfish, and untreated drinking water. Even just one drop of infected liquid has enough bacteria to infect a human, which then they will become a carrier. Both animals and humans can be carriers, and infection of food is very common when cross-contaminated or handled improperly. By consuming contaminated food is the most common way to contract this bacterial illness. Symptoms of Campylobacteriosis are very similar to flu-like symptoms, but can lead to long-lasting effects to one's health. The most common symptoms include diarrhea, abdominal pain, high fever, nausea, vomiting, and the worst symptom, no symptoms at all. Being asymptomatic is dangerous because if you have contracted the bacteria, you now are a carrier and now are spreading the bacteria while handling food, potentially putting others at risk. However, being asymptomatic is uncommon. It is still a very serious issue to get under control as fast as possible. Symptoms typically develop 2 to 5 days after coming in contact with the bacteria, though in some rare cases, a person can become infected and only have symptoms appear a month after the initial infection. This poses a hard task to try to pinpoint what had infected the individual. The illness period where an individual is symptomatic can last anywhere from a day to 10 days, and a very common repercussion of this illness is dehydration due to severe loss of fluids through diarrhea and sometimes vomiting. Case studies have shown that children under 5, young adults ages 15 to 19, and individuals over 60 are more susceptible to this illness. However, there's no data to back up why this is so. Although initial symptoms are very mild, exposure to C. jejuni is associated with the development of many long-term health problems. The most common are 1. Guillain-Barré syndrome, which is a neurological immune disorder where the body attacks the peripheral nervous system, causing weak muscles leading to potential paralysis. And 2. Reactive arthritis, also known as Reiter's syndrome, which is an inflammatory arthritis often affecting the digestive or urinary tract. Other more rare post-infection health risks include IBS, septicemia, gallbladder inflammation, chronic colitis, as well as potentially cancer. Fortunately, contracting this bacteria is very easy to prevent. Remember earlier when I had mentioned that C. jejuni is a microaerophilic as well as a non-spore producing? This means there are many ways to kill this bacteria, deeming it ineffective. Many ways that pertain to the average human would be just to practice safe food handling practices when preparing food. Cooking food thoroughly, keeping different food separated from one another, keeping cooked and uncooked food in different containers or prep plates, flipping or even using different cutting boards for raw food and vegetables, buying pasteurized dairy products, and most importantly, if you are infected, not handling or preparing food are many ways to prevent spreading this illness. Though there are ways to prevent it, and it tends to go away without treatment, sometimes antibiotics are necessary for the health of the individual. C. jejuni is growing resistant to certain antibiotics, including fluoroquinolines, macrolides, trimethoprim, and beta-lactam antibiotics that include penicillin and cephalosporins. It is also becoming resistant to tetracycline, quinoline, and kanamycin antibiotics. There is still susceptibility to some drugs, which include macrolides, aminoglycosides, chloromephenicol, nitrofurantoin, gentamicin, and some tetracycline. C. jejuni also can be killed by certain disinfectants. One which is commonly used in our lab which is the 70% ethanol. However, it must be in contact with it for a minimum of 1 minute to kill all bacteria. On to news. Recently, C. jejuni has made it into the news with 2 outbreaks in 2024, one in July in Washington and another in April in New York. This is a result of drinking raw milk, which has become a fad on social media. Many influencers are promoting the consumption of raw milk because its health benefits, while completely disregarding the harms. Many people do not realize foodborne illness harm from consuming raw product, and it has led to contraction of Campylobacteriosis. Another story found in the news recently pertains to us as Canadians. At the Paris 2024 Olympics, several of our Canadian track and cycling athletes contracted Campylobacteriosis while in competition. Zoe Scherer, a 100-meter sprinter, had fell ill before her race and unfortunately came last. Michelle Harrison, a 100-meter hurdler, fell ill with Campylobacteriosis as well, making her first and last Olympic experience terrible. She claims she does not have her energy or nervous system back since having contracted the illness. Campylobacteriosis also affected a number of cyclists who were not staying in the Olympic Village. Although no Canadian athletes missed competition, it definitely put a damper on their results and time spent on the world's largest stage. With some good news, a Campylobacter jejuni specific antibody has been identified by scientists at Osaka Metropolitan University that gives hope to the development of a vaccine. They developed an antibody that is able to identify the bacteria and inhibit growth. With this recent discovery made in September 2024, the group hopes to develop a way to prevent Campylobacter illnesses from getting any more severe, and also formulating a simple way to detect C. jejuni. By being able to detect C. jejuni quickly, it will help prevent sending contaminated foods into stores or restaurants, and this would be extremely beneficial to microbiology. Thank you for watching.